Good morning, everybody. I will be talking about fruit drop. We all know this meeting is focused on fruit drop, and I'm really glad because Fernando introduced some of the things that I would like to discuss in my presentation too. And it's really neat things that Fernando is finding. We are finding something very similar, or you say things that kind of follow the same pattern. So that's always reassuring for us, and hopefully it will be reassuring for you. Monji, in case I have heard previously somebody had complained that my voice was not good in another meeting, so do let me know if you hear something not good.
Sounds good.
Okay, so Fernando already showed this is just fruit and flower cycle. Here you have from basically March through December. In case of Hamlin is where you see all the flowering and fruit activity happening. And during these different stages, you see different kinds of drops. So when you have flowering happening, you do see flower and bud drop. Then you also see fruit drop, fruitlet drop happening when the fruit set starts happening. You have fruit set happening, so at the same time. Then in May, June, sometime in Florida, it usually happens in May. That's pretty much when the fruit are very young and developing. You see June drop happening. And then sometime from October to December, in case of Hamlin's, you start seeing the pre-harvest fruit drop, and you have the harvest. At the same time, there's flower bud induction going on. And the reason why I want to show this is that anytime you have the flower or the fruit, sorry, in the tree, there's something happening for the next season's crop. And that makes our management really difficult because you not only have to think about the fruit drop, you have to think about if you do something, what it's gonna do to your flowers for the next year. So for example, previously to enhance flowering or flower bud induction, we used to induce drought stress at the flower bud induction period. Now we do know that HLB increases pre-harvest fruit drops, so you don't want to induce drought stress at that time. And those type of management questions are really important or becomes very critical, and you have to think about the whole couple of years, or actually you just cannot think about one crop. Now talking about the stages of fruit development, and I am showing this because in my presentation you will see again and again that it is really important to understand how the fruit develops on the tree. And similarly, Fernando had mentioned that you have to think about, for example, in his case, zinc application, when do you want to apply it? And it's really important to understand stages of fruit development. So there are 3 stages of fruit development in citrus. And so starting in March, when you have the fruit set happening, there's stage 1. This is the exponential growth stage. The fruit is increasing in size, or basically there's a lot more cell division going on. So the fruit is growing, but you may not see a lot of growth. Stage 2 is when you start seeing fruit increasing in size. And this is the cell enlargement, and you see a great deal of increase in the fruit size. And then sometime in October, November for Hamlin, December, January for Valencia, when you start seeing basically color break is the stage 3 of development. So stage 3, you see color changes, you see changes in the sugar and the acid in the fruit, but not so much in the fruit size. Fruit size is pretty much done by this point. So stage 1 and 2 is where a lot of things are happening. This is basically the foundation of the fruit. So you need to talk. We will be talking more about this. Now, in all of my research that I'm going to show you, because we don't have any healthy trees anymore, and HLB study has to be done in field with the fruit drop, we divided all the trees into mild, moderate, and severe based on the HLB symptom levels. So a mild tree will be a dense, good-looking tree, and a severe tree is where you have a lot of quick dieback. And that's how we did all the fruit drop study. First thing that we found was that the HLB symptoms, so if the tree is mild, it has a dense canopy, it correlated with the PAR value of that tree. PAR is the photosynthetically active radiation. Basically, it means the amount of light penetrating. So it's what it's telling me. If you have a good canopy, your symptoms are mild for HLB. It makes sense. But from research point of view, what we are finding is here, the graph is showing you shoot dieback in different levels of mild, moderate, and severe trees. We were measuring shoot dieback in the winter months or fall and winter months until spring. The first line here, so the top graph is for Hamlin and Valencia. The top line is for the severe tree. And what we saw is that the severe trees go through a lot of dieback in these months. There is a quite a bit significant dieback of shoots happening. Also, if you think about it, this time period is when you will see Hamlin drop happening in this part. And then in Valencia, you will see drop happening. So we do see a significant dieback happening in these trees, and that dieback is directly related to the HLB severity. This was, we counted the branches that were dying. And also, this means that we have less viable buds for floral development and vegetative growth. So significant dieback in winter months and early spring. On the whole tree level, we again have Hamlin and Valencia. We are still looking at the tree density or the canopy density. And what we saw is this filled dot is for the October readings, and then this diamond shape is for the May reading. So October to May, you see an increase in dieback, and this increase is more significant in the severe trees and then compared to mild trees. In Valencia, we did not see such trend, but we did see severe trees going through more dieback. So on a whole tree basis as well, this dieback is pretty significant. It's almost 50% of increase in the dieback. So your trees do go through a lot of dieback. So it's going to affect your current fruit and then the fruit that is growing on the tree. So that is a big concern with severe trees or with HLB. Now talking about flowering a little bit. So we were trying to answer if HLB affects flowering in citrus trees, and that is because the weather in Florida is, of course, very fluctuating. Therefore, we have multiple waves of bloom. We knew that from healthy trees. But also with HLB trees, we have compromised root system. There is a correlation between water potential and HLB severity. Basically, severe trees have lower water potential, means they are going through more water deficit. And water deficit has an effect on flowering. So does that affect flowering? And we counted or we studied this. So in this graph, we are looking at Hamlin and Valencia again. We have 3 disease levels. The dark color is for the severe trees in both the graphs. And what we found is that severe trees, it's not significant here at 0.05 level, but this is just per branch wise. We do see a little bit of increase in the flowering happening. And maybe we are repeating this study for the whole tree rather than just the branches, but this might be something significant if you look at the whole tree basis rather than just one branch, that more intensity or more flowering happening in HLB trees. So something to consider is if your severe trees flower early or a small portion of flowering is happening in the trees about a month early, potentially more early flowering means early fruit set, and early fruit set will just expedite, these fruit will be ahead of the rest of the fruit. And maybe these fruit contribute to increased drop. And that's something that we are looking into. So there is definitely benefit in synchronizing flowering as much as possible in order to reduce fruit drop. That may not be the only cause of the fruit drop, but it's worth noticing, and maybe you need to look into your growths when you are thinking about fruit drop. That are you seeing more multiple waves of bloom? And if you are, then probably you need to look into what can you do. I'm not going to talk about the GA and flowering here, but gibberellic acid is one of the plant hormones that are labeled for use in citrus, and you can suppress flowering with that. So starting from the early on, we did another small study, and we are repeating this. This study was done last year, and due to COVID, there has been some issues with it. But what we were looking into is all the different times between severe and mild trees when you see fruit drop happening. So in this study, we tagged about 100 open flowers on severe and mild trees at the same time. So they all were tagged around March 23rd. And then after a month, we came back. That's when there was ease in due to pandemic, we could come back to the field. So in April, what we found is that there is a poor fruitlet retention in severe trees compared to mild trees. So this fruit drop in severe trees is actually beginning sometime as early as the fruit set is happening. So yes, we see a lot of pre-harvest drop happening at the end of the year, but there are things happening as early as April that are resulting in poor fruit set or poor fruit retention in severe trees. Like I said, we are repeating this, and hopefully we'll have more information, but this again tells us that we need to start thinking about fruit from the early on stage. Now, talking about the pre-harvest fruit drop here onwards, initially, when I started working on this about 7 years ago, the hypothesis was if the carbohydrate flow is blocked to the fruit, which results in the carbohydrate stress in the mature fruit, resulting in pre-harvest fruit drop. So this is what we were trying to answer. And for that, we have done multiple experiments to understand what is happening. So first and the foremost thing, we did find in Valencia and Hamlin both that the severe trees drop significantly more fruit than mild and moderate trees. We knew that. And this is the graph for Valencia, and the more dramatic increase in the drop starts somewhere about, so if your harvest is supposed to be in April, generally speaking, mid-February is where you start seeing more dramatic increase in the fruit drop. And similarly, this graph is for Hamlin, a similar trend in fruit drop. So if you are generally harvest your fruit in December, sometime in October is when you see more dramatic increase happening in the fruit drop in severe trees. So something happening here, or maybe that's when you see more dramatic increases. So more symptoms you have, more fruit drop. We looked into the amount of photosynthetically active radiation that penetrates to the tree and tried to correlate it with the fruit drop. So again, the PAR value tells us how dense your canopy is. What we found is that the fruit drop rate is directly related with the canopy density. So the thinner the canopy is, which means more light is going through your tree because you don't have as many leaves, more fruit drop you see. So having good canopy is really, really important. Investing in the canopy is one of the most important things that we think that we have found so far. So better canopy is where the investment needs to be. In addition, we did see that the severely symptomatic trees, so these green color bars or these trees, have significantly smaller fruit than mild trees. And it's again same in Hamlin and Valencia, and we have seen it multiple times. So overall, if you harvest all the fruit, your average fruit size is smaller in severe trees. And we do know that since the advent of HLB, the fruit size has decreased. So this kind of makes sense. Then we looked into the attached and the dropped fruit. So attached fruit is the one that stayed on, that was retained on the tree until harvest. And the dropped fruit is the fruit that dropped during 3 or 4 months before in the pre-harvest fruit drop window. And what we did see, so here again, the blue color is for the mild and green is for severe, and these patterned bars are the dropped fruit. And we consistently saw that the dropped fruit is always smaller than the attached fruit. And this is the case in Hamlin, same thing in Valencia. So dropped fruit is always smaller altogether. So what we know so far, severe trees have small fruit, and then always the dropped fruit is always small. So that is interesting. Now, in order to understand what is causing these, what is happening fruit drop wise, or what is causing the fruit drop, we used a method FDF, and Dr. Fernando did mention about it, that a fruit detachment force, basically it tells us which fruit are tight or hanging on tightly on the tree, and the fruit that are loose on the tree. 6 kilogram force is a good cutoff value. Anything below 6 kilogram force means loose fruit. So we harvested the fruit from the severe trees and mild trees, and as expected, we found that there was a higher number of loose fruit on the severe trees, and a small amount of loose fruit on the mild trees. So these are some of the examples. So the fruit that you see in the red square are the ones that were loose. Anyways, so when we looked at the carbohydrate levels in the juice of these fruit, so we did not see any, well, interestingly, what we saw is, let's talk about glucose here. The patterned bar is the bar, or is the fruit that was loose. And what we found that the juice from the loose fruit of severe tree has more glucose than the fruit that stayed on the tree, or compared to the mild fruit. So this is really interesting, because what it is telling us that the sugar starvation is not happening, at least for fructose, glucose, and inositol, the three sugars. The trees are, or the fruit is not starved for these sugars. If anything, loose fruit has more. Now, you may argue, or we have looked into that, we do know that the fruit size is smaller for these fruit, that the loose fruit are smaller in size. So it is possible that the small fruit are more concentrated in sugars. Nonetheless, fruit size is a factor of water availability, or how much water gets into the fruit. The sugar were able to get to the fruit, it's the water that didn't get into the fruit, or the fruit did not size up. That's the problem here. So the carbohydrate starvation is not the cause. In addition, we looked at number of other factors like leaf number, aborted seeds, blotchy mottle, and we didn't see any effect. So only factor that is differentiating loose and tight fruit and is fruit size. Loose fruit or the dropped fruit is always smaller. Now, we wanted to look into how fruit drop progresses over time, because in order to have a good strategy to reduce fruit drop, we need to know what exactly happens over time and when should we apply these strategies. So in this case, we measured the fruit detachment force on Hamlin in October and in November. As expected, we did see that the number of loose fruit, which is the blue color here, it increased over time, and not so much in the mild trees. So severe trees were having more loose fruit over time, expected. Then we looked at the gene level or the molecular analysis basically to see what is exactly happening. So next couple of slides have these graphs. What you're seeing here is each panel is a gene, and it's a gene related to fruit drop in one way or another. Here, these genes are the cell wall remodeling gene. This basically means if a fruit has to detach from the tree, there has to be something happening in the cell wall for that will result or that will ease in this detachment or the drop of the fruit. So in these slides, the first row is fruit collected in October. The bottom row is the fruit collected in November. Now, if you remember, or you probably already know, you see more drop happening in November. That's when you have more loose fruit on the tree and you see more fruit on the ground. October, not so much. When we looked at these genes, what we found that there is more activity of these cell wall remodeling genes happening in October, not so much in November. There's more things happening in October. Then we looked at ethylene biosynthesis and ethylene signaling. Ethylene accelerates fruit drop. And interestingly, here also we saw that there is more activity of ethylene-related activity happening in the loose fruit in October and not so much in November. So a lot of these things are happening in October when you don't see so much fruit drop happening. Then abscisic acid biosynthesis related gene. This gene helps with abscisic acid. Abscisic acid is another plant growth regulator. Fernando had mentioned about it. Basically, abscisic acid is known to increase once the abscisic acid level increases. There is an increase in production of ethylene, and ethylene can cause abscission. And what we did see here is that there was more stuff happening again in October. This ABA biosynthesis was, or the gene was at least more expressed in October, not so much in November. This is for Hamlin. And we again saw the same thing for Valencia, that loose fruit had higher expression for ABA biosynthesis in February. So this is Valencia. That's why we are talking about February, and not so much in May when you actually see a lot more fruit on the ground. So now talking about water potential, because abscisic acid is also expressed in, or abscisic acid is basically produced in response to drought stress. Whenever your tree is experiencing drought stress, there is enhanced production of abscisic acid. So we measured leaf water potential. This is in case of Valencia. And what we found when we measured leaf water potential in May, which is the time of harvest in our case, we did not see any differences in the water potential at this time point. So that was interesting. In another experiment in Valencia, we had measured leaf water potential in March. And that's when we saw the differences. So this mild tree was not experiencing as much water deficit as the severe tree. So that is very interesting because we saw abscisic acid related genes being expressed more in February or 2 or 3 months before the pre-harvest drop. And the leaf water potential or the water deficit is also a little bit early, not at the time when you see the drop. Then we do know that HLB affected trees have smaller root biomass, so water nutrient uptake is limited in these trees. Now talking again about the fruit size. So here in this graph, what you're seeing is the blue color is for the mild tree and the red color is for the severe tree. We measured the fruit size throughout the year. And in the beginning, you don't see so much differences in the fruit size. It's basically around June. So stage 1 or 2 of fruit development is when you start seeing these differences in the fruit size, and they become more and more obvious as the time progresses. To remind you, this is stage. We are talking about somewhere here when we start seeing differences in the cell size. And this increase in size is driven by water availability. So this is another data set, another experiment, but same thing as earlier. Maybe we are seeing differences in fruit size. So fruit size is driven by water availability, and typically April and May are dry periods. So perhaps the water deficit that happens in the early time of the fruit set, that is sufficient to make severe fruit lag a little bit in fruit size. And we do know HLB affected trees have small root biomass. Severe trees have even smaller root biomass. Evan Johnson's research has shown up to 80 or 70% root loss can happen in severe trees. So that is something to consider. So this is what we found, how the fruit drop is progressing. We also did another study where we were trying to see what may be causing fruit drop. And in this case, we compared SugarBelle, which is a mandarin, to sweet oranges. We do know SugarBelle doesn't drop as much fruit. This is just some data to support it. We compared it to mid-sweet and to Hamlin. And this is basically, we left the fruit on the tree when rest of the grove was harvested. So these many weeks after the harvest has already been performed on rest of the trees. SugarBelle did not drop more than 20% of the fruit, whereas a mid-sweet was dropping about 60% and Hamlin was dropping 40%. So we have data to show that SugarBelle doesn't drop as much fruit. So we wanted to compare it to sweet orange, Hamlin, and see what are the key differences between SugarBelle and sweet orange that causes this difference, that SugarBelle doesn't drop fruit, but Hamlin does. So we did in-depth molecular analysis, and these are some of the key hormones that we know that we found to be differentially expressed between these SugarBelle and Hamlin. And what we found that in case of Hamlin, we saw increase in genes related to ethylene biosynthesis and signaling, increase in ABA. Then we also saw increase in strigolactone and cytokinin. And then the genes auxin, which is usually helped with the tree fruit growth, we saw a reduction in auxin and a reduction in GA. So what it all together means, and then we also found some oxidative stress, but altogether what this study showed us is that changes in the hormone homeostasis is happening. So we see a lot of increase in the negative regulators of or the positive regulators of abscission. We also see a lot of increase in the oxidative stress, and we did see lots of hormone genes related to oxidative stress. So HLB is causing changes in the hormone, increasing the oxidative stress, which results in cell wall modification, resulting in abscission. So altogether, it's hormone and oxidative stress that is resulting in the increase in the drop. So this is so far what we have learned about the drop. Now, moving to a GA study that I have presented a couple of times now, but I want to bring it again to your attention as one of the possible strategies that you can use. So we were conducting this study. It was focused flowering study, but we found something interesting from the fruit drop point of view. So in this study, we were applying GA at 20 gram active ingredient per acre from September to January. First thing that we found that with the use of GA, we saw an increase in the fruit size. Fruit size was always larger for the GA treated trees. And when we looked at the fruit detachment force, it was higher for the GA trees as compared to the control, which means that the fruit removal was difficult on GA tree. And we did see a positive correlation between the fruit detachment force and fruit size. So larger the fruit is, more difficult it is to remove from the tree. So all of that indicates that GA is helping with the fruit retention and fruit size improvement. When we looked at the yield response, this study was started in 2017. Now it has been going on for 4 years. And overall, every year we do see an increase in the GA treated trees. It was cumulatively, we do see an increase 2017 and 2018, although it seems a little bit increased, but there was no statistical differences. But 2019 is when we do see a statistical difference, and 2020 as well, because now we don't see as much variability in the trees. So we are seeing increase in the pounds per tree with the GA application. So just interestingly, GA does reduce flowering, but we are seeing increase in the yield. So we have fewer flowers to begin with, but the yield is increased. Here is a correlation analysis in the same trial. We have different GA application treatments too. So I'm only showing you the results of GA 5-time application, but we have some other treatments. And the interesting thing that we found is that the more GA you apply, there's a positive correlation in the yield. It's a mild relationship, but 0 GA, as you go from 0 to 100 GA, we are seeing increase in the yield. So GA seems to be promising in improving our yields. So we did a small molecular study again to see what is happening, what may be causing this improved tree health and improved yield. So PP2, CDR1, and LOX2 are some of the genes that we know are related to HLB. And we did see higher expression of CDR1 and lower expression of LOX2 in the HLB, in the GA treatment, suggesting that maybe GA is helping the tree in improving tree health, and is they helping in fighting infection. Somehow it's improving the plant defense response. So overall, GA does hold a potential to improve fruit set and fruit retention. We have multiple years of data to prove that. And we are doing more research to identify the time, more specific time of application, because all of this work was done September to January application. With that, I will end my presentation. The take-home messages that I have is that HLB triggered events that lead to abscission occurs earlier in the pre-harvest period. So when you actually see the drop happening, it's too late. The events are happening way sooner than that. Good canopy is equals to less drop. Small size fruit are more likely to drop. And increase in fruit drop is not like due to starvation of carbohydrate. Increased oxidative stress caused due to disease, it seems to help improve or causing more drop. And abscisic acid seems to be involved in triggering fruit drop. I'm sorry, my bottom bullet point cannot be seen, but what I'm trying to say is good caretaking is what is important. You need to have good irrigation plan, small doses of water, small and frequent irrigation, starting from the early April when you see dry periods, March, April, May, when we really have dry period. Having good irrigation plan at that time would help in your fruit size, which eventually will help in reducing fruit drop and having better fruit quality. So with that, I will end my presentation. I do want to thank Citrus Initiative because most of the work, majority of the work is funded by that. Valencia Biosciences for providing us GA. CRDF was the funding source for the GA trial that was focused on flowering, and Peace River Packing has been our long-term collaborator. And my lab, it's all of their hard work. So thank you.